Corneal dystrophies. These are bilateral corneal changes that occur spontaneously, mostly in second decade of life. They are genetically inherited, mostly as autosomal dominant disorders, and they could be stationary or slowly progressive. Corneal dystrophies develop in absence of inflammation or systemic diseases. Genetics. Cells contain nucleus with chromosomes. Chromosomes are classified into 46 chromosomes divided into 22 pairs of autosomes, 1 pair of sex chromosomes, XX for females, XY for males. Each chromosome is formed of a short arm and a long arm. Both arms join at centromere. According to overall morphology, chromosomes are divided into groups labeled A to G. Chromosomes are made of supercoils of DNA, euchromatin, light stained with active genes, heterochromatin, inactive, unexpressed, repetitive DNA. Deoxyribonucleic acid or DNA. This is formed of 2 complementary strands of nucleotides. Double helix, hydrogen bonds occur between bases of nucleotides. Nucleotide is composed of the following: phosphate molecule, sugar molecule, nitrogenous base. Purines as adenine and guanosine, or pyrimidines as cytosine and thymine. Pairing occurs as follows: guanosine with cytosine and adenine with thymine. 3 billion pairs of bases make up human DNA. Codon. Codon is a group of 3 nucleotides specifying certain amino acids. These are the smallest segments of DNA. They contain individual units of information coding for a single trait or a single polypeptide chain. Genetics and corneal dystrophies. Genes on at least 9 human chromosomes are involved in maintaining corneal transparency, including chromosomes 1, 5, 9, 10, 12, 16, 17, 20, and X. Specific genetic mutations in 6 genes have been identified in corneal dystrophies. Keratin genes, Meesmann's dystrophy, beta-transforming growth factor-induced gene, BIGH3, granular, lattice, Avellino, and Reese-Buckler. Gelsolin gene, lattice dystrophy with systemic amyloidosis. Gastrointestinal tumor-associated antigen gene, M1S1 gene, familial subepithelial corneal amyloidosis. Steroid sulfatase gene, cornea farinata and deep filiform dystrophies. Epithelial and subepithelial dystrophies. Meesmann's dystrophy. This is rare bilateral autosomal dominant dystrophy appearing very early in life. There is inherited defect in corneal epithelial specific cytokeratins K3 and K12. Mutations result in epithelial fragility. KRT3 gene mutation, 12q13 locus. Mutation in codon 509 where glutamic acid is replaced by lysine is present. KRT12 gene mutation, 17q12 locus. Several mutations within the gene. Arginine 135 glycine, arginine 135 threonine, tyrosine 429 aspargine. Meesmann's dystrophy has an early onset, 1 to 2 years of life. There are recurrent erosions with good vision. Small punctate intraepithelial vacuoles are seen. Microscopically, there are abnormal epithelial cells containing cytoskeletal peculiar substance. Thickened epithelial basement membrane with increased epithelial fragility. Dot and fingerprint dystrophy. Anterior basement membrane dystrophy. This is the most common anterior corneal dystrophy with poor epithelial adhesion, turnover, maturation, and production of basement membrane. It is an autosomal dominant condition affecting 6 to 18% of population, most commonly women above 50 years. There is poor epithelial adhesion with microcyst and bulla formation. epithelial reduplication and/or folding, excess sub- or intraepithelial production of basement membrane material and collagen, thickened basement membrane, recurrent epithelial erosions. Subtypes of map dot and fingerprint dystrophy. They often coexist. Fingerprint. Parallel relucent lines of basement membrane separating tongues of reduplicated epithelium. Map. Geographic. Subepithelial connective tissue resembling degenerative pannus. Microcystic. White, putty-like contents reflect degenerated epithelial cells trapped in disorderly epithelium. Reis-Bachler's dystrophy. Progressive autosomal dominant Bowman's layer dystrophy. There is mutation in chromosome 5q31 locus. Mutation in beta-transforming growth factor-induced gene, or BIGH3 gene. Protein product of BIGH3 gene, keratoepithylene. Gene is normally expressed in corneal epithelium. There is missense mutation, causing a single amino acid substitution in the gene's protein products. It occurs in the first decade of life with recurrent erosions. Superficial central geographic, gray-white, ring-shaped central opacity is seen. Microscopically there is irregular saw-toothed epithelium, subepithelial connective tissue scarring, destruction of Bowman's layer, laminated pannus, eosinophilic crystalloids that stain red with mesenteric.
Primary gelatinous drop-like dystrophy, familial subepithelial amyloidosis, amyloid composed of mutant lactoferrin. Stromal dystrophies, granular dystrophy, Gronow type 1, Bachler's type 1. It's an autosomal dominant stromal dystrophy. Etiology, produced by epithelium BIGH3 mutation. Mutant keratoepithelium forms granules. Pathology, course, late onset, slow progression, visual loss is late, and can recur in graft.
There is bilateral central superficial ring, or crumb-like opacities with clear corneal periphery. Microscopically, there are hyaline rock candy stromal deposits that are intensely red with mesenteric acid phosphonophilia. The origin is non-collagenous protein derived from corneal epithelium or keratocytes. By TEM, electron-dense granules with periodicity appear. Lattice dystrophy, type 1, by Ihab, Bachler's type 3. This is a common autosomal dominant primary amyloid deposition in anterior stroma. The onset is in the first decade. It is a bilateral condition with BIGH3 mutation. Mutant keratoepithylene forms amyloid. It is a form of localized corneal amyloidosis. Penetrating keratoplasty is usually necessary in the 4th or 5th decade. Pathology. Gross picture. Delicate branching relucent lines in stroma, recurrent erosions, and superficial scarring as in Reis-Bachler's. Microscopic picture. Intrastromal and subepithelial deposits of amyloid. Amyloid stains Congo red. There is positive apple green birefringence and dichroism with polarization microscopy. Abnormal overlying epithelium, atrophy or hypertrophy. The dystrophy can recur in graft. Macular dystrophy, localized corneal mucopolysaccharidosis. This is a rare autosomal recessive form of stromal dystrophy involving corneal periphery. It is the most severe, visually disabling. Deposits are glycosaminoglycans, acid mucopolysaccharides. There is defective sulfonation of keratin sulfate molecules, proposed type 1 enzyme defect. Insoluble non-sulfated keratin, sulfate, accumulates in keratocytes, endothelium, and between stromal lamellae. There is abnormal stromal hydration. Macular dystrophy has an early onset, progressive course, and needs penetrating keratoplasty in 3rd decade. There are superficial opacities with indistinct borders that begin axially, and there is diffuse stromal haze between opacities. Microscopically, the corneal stroma is not thickened. Glycosaminoglycans are basophilic deposits which stain with colloidal iron stain or alkyne blue stain for mucopolysaccharides. Mucopolysaccharides are present in keratocytes, endothelial cells, and stroma. Macular dystrophies can recur post-keratoplasty in corneal transplants. Schneider's central stromal crystalline dystrophy. This is a primary disorder of lipid metabolism with cholesterol deposits in corneal stroma. It is autosomal dominant, associated with systemic lipids, xanthelasma, elevated serum lipids. It may necessitate penetrating keratoplasty by the age of 40 to 50. There is diffuse central stromal clouding. Microscopically, there are needle-shaped polychromatic cholesterol crystals in anterior stroma, prominent bilateral arcus. It stains with oil red O stain. Fuchs combined dystrophy. Cornea guttata, primary endothelial dystrophy, adult onset. It is a primary endothelial dystrophy with secondary changes in the epithelium. It is autosomal dominant, affecting the elderly female bilaterally, and it runs a progressive course. Microscopically, there is guttate excrescences of abnormal basement membrane material secreted on Descemet's membrane. Descemet's membrane is thickened, often multilaminar. Guttae may be buried by retrocorneal membrane. Pigment phagocytosed by endothelium. Secondary stromal edema and bullous keratopathy are present. Congenital hereditary endothelial dystrophy. There is diffuse blue-white opacity affecting neonates. It is a rare disorder, autosomal recessive but rarely autosomal dominant. Its onset is at birth and it is bilateral and progressive. Microscopically, there is thickened edematous stroma. Stromal collagen is separated by fluid clefts. Massively thickened Descemet's membrane, atrophic or non-functioning endothelium.